Okay, hi everyone. Welcome to my presentation this afternoon. And so I'll be basically talking about the development of the Karman database, which is a proteogenomic database that we hope will in some way advance the development of and the validation and identification of novel cancer vaccine as well as immunotherapy candidates. So, okay, so that is the overall sequential flow of my presentation. So we're just going to be building up towards the middle of it, which is the development of the database and where we want to go from there. So just to put things into their proper perspective, so when we when we talk about immunotherapy today in general and immune checkpoint inhibitors in particular, so we tend to think of it as something of a recent development, but that's not actually true because the basic science behind immunotherapy was actually almost 60 years in development. So if we focus on like the first half of the slide, so we can tell that, the 20th century was focused on developing the basic science and the principles that led to, the development of these immunotherapies and immune checkpoint inhibitors, and the 21st century, the 2010s onwards. So they started with the translation of those, basic scientific principles into a clinical setting and giving us the checkpoint inhibitors that we use today. So this slide basically encapsulates the fact that, 1953 or 1950s is, where it all started, and 2022, last year, that's where we are right now. And that's the point at which, we're going to continue. When we talk about immune checkpoint inhibitors in particular, so what we know so far is that immune checkpoint inhibitors target a diversity of cancers. So there's a whole host of cancers which today can have immune checkpoint inhibitors that have been approved for their treatment. And the 2010, so 2011 onwards, so it saw a spurt in the growth of approvals for immune checkpoint inhibitors. So what we also observe is that most of the immune checkpoint inhibitors that have been approved, they tend to focus largely on 4 or 5 types of cancer. So melanoma, non-squamous cell lung carcinoma, renal cell carcinoma, etc. And despite the fact that, there's a whole lot of other cancers that have also been targeted by immunotherapy. When we look at cancer vaccines, on the contrary, we find that compared to immunotherapies, compared to checkpoint inhibitors, the diversity and the rate of approval for these cancer vaccines is significantly less. I mean, there's barely like 4 or 5 cancers that can actually be targeted by cancer vaccines, basically. And most of those cancer vaccines happen to be preventive, not therapeutic. So, if it was therapeutic, then you would just treat the cancer with the vaccine. But preventive vaccine, it's to prevent the cancer from happening. But, so there hasn't really been a lot of progress in terms of cancer vaccine approval as of today. And this is, in fact, where resources such as ours could potentially have an impact, because what's holding us back is essentially the lack of, targets. We have the data. We've analyzed, data from multiple cancers, but we just don't have the targets. And to get the targets, we really need probably to analyze deeper and at a higher resolution, and we have to target as many cancers as possible. So this is hopefully where resources such as ours could make a difference. And there's also a problem with, immune checkpoint inhibitors, because they don't always work, and I mean, they're very effective. They've totally revolutionized, cancer treatment in the past decade or so. But for many patients, they just don't work. And what we see here is some of the reasons, why they don't work. So firstly, there is the problem of treatment resistance. So depending upon at what phase of the treatment, you develop resistance towards. So it could be primary or acquired resistance. And then there's a problem of treatment-related adverse events. So, what we see here are three of the most common treatment-related adverse events that the patients have experienced. And some patients actually experience death, but death is not a very, fortunately, it's a very rare adverse event, but it still happens with immune checkpoint inhibitors. And then there's also the problem of, and this is quite a significant problem, the lack of T-cell infiltration into tumor tissue. So basically, I mean, you don't see T cells in the tumor tissue. They're not infiltrating. They're not getting inside. So there's no immune response. And a patient who has a symptom like that, who doesn't show T cell infiltration, his immune checkpoint inhibitor therapy is just not working. So these are some of the reasons why this therapy doesn't work. And therefore, I mean, this is probably an area of where resources such as ours and others like ours could probably contribute in terms of novel target discovery and developing new routes or pathways for treatment for different patients. And one of the sources for new target discovery is actually immunopeptidomic mass spectrometry. So immunopeptidomic mass spectrometry is basically a subset of proteomic mass spectrometry. And in immunopeptidomic mass spectrometry, what we are looking at is the short peptides presented on the surface of different cells, and those peptides which are then surveyed by the HLA complex, the human leukocyte antigen complex, which therefore initiates an immune response in the patient. So immunopeptidomic mass spectrometry is actually combined with methods such as sequencing. So from mass spectrometry, you get the peptide sequence. From sequencing, you get yourself the mutations and similar information about those peptides. So at the end of it, so you have like a combined proteomic as well as genomic, you have data from both resources. So you use that data to be able to identify the mutations in the patient and the peptides which are showing those mutations. And those peptides could then be used for developing, they could be used for identifying new targets and developing cancer vaccines and novel immunotherapies. So neoantigen identification and discovery is obviously the driving force behind the development of the Carmen database. And how we get there is shown in these three stages. So there's the first stage, of course, is identifying the peptides, developing the database, and making it as comprehensive as possible. Stage 2 is about taking those peptides, the genomic information, and identifying regions of the genome which are disproportionate in terms of the antigen presentation. And the third stage would be taking those regions and asking questions of clinical significance. So could those regions of antigen presentation probably predict response to immunotherapy? Okay. And so what does Carmen look like, and what is Carmen basically? So Carmen is basically a database of MHC class 1 associated peptides taken from both healthy as well as cancer tissues. And the name Carmen basically is an acronym for cancer immunopeptidogenomics, because those are the two key, that's basically what defines our work. So it's cancer focused, and it's also a combination of immunopeptidomics and proteogenomic analysis. So, and this is basically what the database looks like in terms of number of peptides, number of spectra, number of samples, etc. And how did we actually get to this stage? How did we develop the database? So the raw data that we have for the database came from two major resources. So that's basically Pride and Massive, which are the two most widely used proteomic resources online. And so with a series of pipelines where the data was converted, it was transformed, it was cleaned, it was then fed into a snake-make-based pipeline known as COD-DIP. So COD-DIP is basically an acronym for the three types of database searches that are performed. So closed, open, and de novo. And each type of search basically identifies a certain type of peptide. And the de novo peptide is an interesting class because those are the peptides which don't currently exist in any of the proteomic databases, such as UniProt and Ensembler, for example. So those peptides could possibly be novel. And so after identification of those peptides, we then derive additional features of those peptides, like their binding affinity. We map those peptides back to their genomic locations, and we also mine the mutations associated with those peptides. And the final result after filtering is what we get is the final version of the database. And well, that is the metadata associated with the database in terms of the fields, the type of data, and an example of the data in each type. So we have 20 fields, and each of those 20 fields has a very specific use case and application. And coming to some of the key results. So firstly, what we see here is an overview in terms of the types of cancers represented. So we have 16 types of cancers represented in our database. We have a mixture of samples which were cell culture, which were tissues, and which were a mix of cell cultures and tissues. And we have also a mix of cancer as well as normal data sets in our database. So this basically talks about what it looks like in terms of those. Coming to some of the important physicochemical and binding attributes of our peptides. So if you look at this figure, so the first thing is that the peptides, they basically they adhere to the common MHC class 1 length distribution. So it's 9 amino acids, and most of them are 9 amino acids. Secondly, we see that there's a diversity of HLA alleles to which the peptides bind. I haven't shown you all of them because there are almost 453 diverse HLA alleles in our data set. So we just looked at the top 20 over here. And we see that the same peptide binds to multiple different HLA alleles, and it's vice versa. Thirdly, I mean, the peptides, the common to cancer and normals, there is some overlap, but there's not a lot of overlap. So that's actually helpful to us going forward. And in terms of the binding affinity, so the number of strong binders that you see in cancer samples and normal samples, they're high in both of them, but they're significantly higher in the cancer samples compared to the normal samples. So we use these features to basically build up towards, prioritizing the neoantigens, because we cannot say that, based only on this, that a peptide will be a neoantigen, because that would be insufficient. But we use these measures, to increase the confidence of calling a neoantigen, basically. And just to have a brief look at the de novo, as I mentioned, the de novo peptidomic landscape, because these peptides as I mentioned before, they're not actually found in any of the proteomic databases, so they could be novel. So we see that they come from a diversity of sources. So we see that they originate from out of frame translation events, from frame shift mutations, from intron exon junctions, etc., etc. And we also see that there's the types of reading frames that are giving rise to these peptides. So we've shown you the top 5 reading frames that could be contributing towards these peptides. And it's important for us to actually be able to explain where these peptides are coming from. So if you look on the far right, so for most of the cancers, for most of the tissues, and for most of the samples, we are able to explain the origin of these de novo peptides. And there's actually a minority of peptides which cannot be explained right now. But it's good because this, again, increases the confidence with which we call a de novo peptide, because we need to be able to distinguish between a genuine de novo peptide and a noisy signal, for example. So, and well, the other thing about de novo peptides and the identification of a de novo peptide in a database is that we need to be able to distinguish between normal tissues and cancerous tissues, because we need to be able to prove that in terms of expression, the de novo peptide is actually being expressed at a level that could possibly be acceptable, and could possibly be something that the wider community could accept as a genuine signal. So to do that, so there's 2 ways to do that. The first thing is to compare the expression of our peptides at the TPM level using the GTEx database, and the other thing is to be able to set a cutoff at below which or above which the de novo peptide expression can be trusted. So what we see here is that most of our de novo peptides aren't actually shared between cancer and normal tissue, and they're mostly exclusive to cancerous tissue, and there's some of them and there's which are exclusive to different cancers. And in terms of expression, the de novo peptides themselves are expressed in very low amounts in cancer, and they're expressed in much higher amounts, those that are common to cancer and normal, for that I mean. And so that basically, I'm sorry, that basically these are the measures that we use to basically increase the confidence with which we well call a de novo peptide. And in terms of the number of peptides common between our database and other published resources right now, so what we observe is that we actually have a lot more unique peptides, de novo peptides, in our database compared to the existing resources. And we also see that we are able to actually call the peptide prism is actually another program that also calls de novo peptides. So if you compare both of them, we can see that we are actually able to call more de novo peptides compared to the existing resources. And lastly, okay, this is a work in development. So we, as I said, we look for regions of disproportionate antigen presentation on the genome. And why that is important is because, as you can see here, so we've zoomed into one gene, basically. So we've zoomed into TP53, but we will obviously, as time progresses, zoom in and look at the trends in pretty much every single chromosome in the genome. And what we observe here is that those peaks that you can see there, those peaks are basically the regions where antigen presentation was found to be disproportionate relative to other regions of the genome. And if you focus on the box over there, so we see that there's those little black circles, basically, those basically correspond to the peaks that you see on top. And if you compare, if you look at the peaks in terms of expression values, in terms of the number of mutations, in terms of population coverage, what we see is that they basically score very highly on the immune score in all 3 counts, which probably means that because of the number of mutations, because of the population coverage, because of the expression, these could potentially have therapeutic significance. So the way forward, basically going from here, is that we complete the development of the genomic model. We integrate that with, we develop a large language model for immunotherapy response, and we integrate that with a clinical trial data that is available to us from our project partners. And yes, and this is the source publication, which is the foundation for my work. And yes, thank you very much.
Thank you. We have time for a brief question, if there is any brief questions. Can you please come down if you have a question?
Thank you for your work and your talk. I was wondering whether your work shares novel insights into the filtering strategies in order of the minimal level of expression, and whether you think it will expand with novel immunoinformatics approaches with the B-cell and T-cell presentation strategies of your...
Okay, so we don't actually look at B cells right now. So our focus is on exclusively on, T cells mediated response. But we haven't looked at B cells because our group focuses on MHC class 1 and T cell mediated responses. So I'm not really sure about that, if it would work.